Last time we left off talking about the innate immune system, right? All the cells that basically makes up the innate immune system, inducing things like inflammation and stuff like that, right? The neutrophils invade in there first, eat up all those pathogens, secretes the nets, and then they die via apoptosis, forming pus. And then the macrophages come in to clean up all of that, right? So the process of both of those type of white blood cells leaving the circulation to move into the infected site of the body, we call that collectively diapedesis. Okay? Another name for that is extravasation. You guys need to know the steps that's involved of how these these particular white blood cells leave the circulation to get into whatever tissue they need to get into. Okay. All right. The next thing that the innate system is responsible for is the production of what we call fever. Now, fever is basically just resetting of your internal thermostat. The internal thermostat regulates the body temperature at a normal physiological range, right? Anywhere between 36.5 to about 37.5 degrees Celsius or 97.7 to 99.5 degrees Fahrenheit, okay? Now, when you're infected with something, and especially if it's like a gram-negative bacteria, as your white blood cells eat up those gram-negative bacteria or any kind of bacteria for that instance, they will stimulate the breaking down of that bacteria, and through that stimulus, it triggers the white blood cells to release these signals. Collectively, these signals work together. Separately, they have their own functions too, as we talk about the immune system and how these particular signals work individually. But all together, these three type of cytokines or signaling compounds secreted by these white blood cells work together to travel together to the hypothalamus, which is basically where your internal thermostat is, and then reset the hypothalamus to a higher temperature. Okay, so these particular cytokines are collectively known as your pyrogens. Okay. And the specific ones that induces this fever production is called interleukin 1 or IL-1, interleukin 6, IL-6, and tumor necrosis factor alpha. Okay, so those three secreted by your white blood cell together stimulates your hypothalamus to reset the internal thermostat to a higher temperature. And once the hypothalamus resets that temperature, it goes up above 37.8 degrees Celsius or 100.5 degrees Fahrenheit. And now this is the beginning of the production of fever. Okay. Now fever is actually quite important in terms of how it can help the body fight off the microorganisms. One of the benefits of fever is to reduce the growth rate of these microorganisms. Okay. Because oftentimes the microorganism, bacteria, whatever it is that invade our body, loves to grow within body heat temperature. Okay, so it grows best within that area. So therefore, if you raise the internal thermostat to a slightly higher temperature, it makes everybody uncomfortable, right? And especially these microorganisms, so they may not grow as well. Now, with the increase in temperature, it may also inactivate a certain amount of toxins that are secreted by some of these bacteria or microorganisms, right? It also, this slight increase in temperature can also increase the metabolism and enhance the immune response. Makes your white blood cells a little more responsive, makes them, a little more triggered so they can go out there and start to eat up a lot more things, so therefore increase phagocytosis. But most importantly, fever also induces drowsiness in the infected individual. So therefore, when an individual is basically going through this particular inflammatory reaction and they produce fever, they tend to, go to sleep, right? So when you sleep, you don't have to worry about those symptoms, you don't have to fight off those symptoms, and you just let your body do its job. Now, of course, fever is beneficial. However, very high fever can have permanent negative effect, okay? Not only on the microorganisms, but also on our body as well. Because you guys understand that enzymes and proteins within our bodies and within our cells work very well within a certain temperature and temperature range, right? So therefore, if you start to change the internal temperature to a much higher temperature, some of those proteins will become denatured, okay? I mean, of course, the microorganisms, the proteins of the microorganisms also become denatured, but our proteins may also become affected. Now, if these proteins are vital for the functions and survival of the cell, then the cell may also die based on the fact that you've increased that temperature to a much, much higher temperature, right? So if you have fever of like, let's say 101, 102 degrees, it's not a big deal, right? Once your fever gets up to about 104 degrees, 105 degrees, depends how long it lasts. If it lasts for more than an hour, then maybe it gets super uncomfortable. So therefore, you may want to take some kind of medication to kind of help reduce that fever a little bit lower. It's just more manageable way, so then you don't kind of destroy your own proteins as well. Okay, so keep that in mind. If you do have kids, and your kids, let's say after vaccination, or when the kid has an infection and the kid has fever, it depends on the age of the kid. If the kid is more older than 2 years old, you can give like children Motrin or children Tylenol, whatever it is, right? If the child is less than 2 years old, there's like herbal remedy that you can give them, or you can just, if the kid has fever, all you have to do is take the kid's clothes off and leave him there. Okay, that will naturally reduce his fever. Okay, because the kid, these children, these 2 years old and below, they have a lot of what we call brown adipose tissue, which constantly goes through metabolism and releases heat, so it'll keep their body warm no matter what. Okay, and besides, they have fever, so you want the environment to help cool these kids down, because you can't really give any kind of medication to kind of reduce that fever. Okay, so keep that in mind. When my kid was sick, when he was like, when he was born, he was sick for like 11 months of the first 12 months of his life, right? I was sick for 11 months for the first 12 months of my life too, or of his life, not my life. But anyway, every time he had a fever, I just take his clothes off, diapers on, and just lay him in the middle of the bed. Even in the middle of winter, he'll be fine. But then my job is have to stay up all the time, right? Stay up for the whole night just to like make sure that he doesn't become hypothermic, right? So I take his clothes off, that the temperature goes down, wrap him up in a blanket again. If it heats up again, take it off. And then if it cools down, wrap it up. Back and forth we go. And then of course, if you're a parent and your kid is sick, you get virtually no rest whatsoever, right? So it's one of the things to kind of look forward to if you guys don't have kids already. Yeah. All right, so that's fever. Okay, another way for the innate activities to really enhance the immune activities of your cells is to activate what we call the complement system. The complement system is a series of 9 different proteins, all made by the liver as well. So all of these 9 proteins will enhance or complement the activities of your innate immune system. Now there's many, many ways to activate these 9 proteins, but the best way to do so is to have what we call antibodies, which are released by your adaptive immune system. And those antibodies will help fight off these particular pathogens, or will help enhance the activities of your innate cells. Okay? So multiple ways for it to work. Overall, the function of the complement system is to enhance phagocytosis, enhance the eating activities of your white blood cells, induce lysis of the microorganisms, whatever the case is, they poke holes on the surface of those microorganisms, and also to induce inflammation and enhance the overall immune responses. Okay? So multiple ways for it to work. Here's 3 different pathways to activate the complement activation. So classical pathway, alternative pathway versus lectin pathway, right? You guys don't really need to know these pathways, but I'm just gonna show you the classical pathway real quick. Okay? So for the classical pathway, this is the only pathway for the complement activation that requires antibodies. Now remember, antibodies are released by the plasma cells, right? We talked about that last time. The plasma cells are active B cells, and the B cells belong to your specific adaptive immunity, okay? So therefore, you do need that component from your adaptive immunity so that you can enhance or activate these complements so that you can enhance the overall activities of your innate immune cells, okay? Neutrophils, macrophages, and whatnot, right? So therefore, to start the classical pathway of activation, you have these 2 type of antibodies called the IgG. So it's a class of antibodies, right, called the IgG. The IgG will bind to the surface bacteria, surface proteins on bacteria. Once the IgG binds to the surface proteins on the bacteria, there's the first complement called the C1 protein. C1 then binds to the 2 IgG, and together they form a complex. That complex is referred to as C1s esterase, which is an enzyme. C1s esterase is going to take C2 and C4 and cut both of those into their subunits. So C2 becomes C2A and C2B, C4 becomes C4A and C4B. Okay? C2A and C4A C2A and C4B combines together to form another complex we call C3 convertase, which is an enzyme. C3 convertase takes C3 and splits it into C3A and C3B. C3B and C4A combine to form another complex called C5 convertase. C5 convertase is going to split C5 into C5A and C5B. Okay? So once all of that has done, now C3A and C5A, they have receptors on the mast cells. You guys remember the mast cells? We talked about the fact that they live outside of the blood vessel, sitting around the blood vessels and therefore releasing substances to induce vasodilation of the blood vessels, right? So therefore on the mast cells, there are these receptors for C3A and C5A. So both of those subunits bind to the mast cells and stimulate the mast cell to release the vast majority of their histamine. Okay? So when histamine is released massively from the mast cell, it stimulates the smooth muscle around multiple vessels and induces massive vasodilation. Okay? Because you guys remember, dilation of the blood vessel enhances blood flow to whatever parts of the body that's infected. So therefore, this enhances even more the amount of blood that breaches the infected site of the body. So essentially, with C3A and C5A binding to the mast cell, it increases the inflammatory response of the body. Okay? C5A also individually acts as a chemotactic compound. So it induces chemotaxis and therefore attract all the neutrophils and monocytes to that infected area. You guys remember we talked about that, right? One of the cytokines released by the resident macrophage very early on in that particular infection. Okay? Now with C5b is going to bind to the surface of your bacteria, of your microorganism, and together it will activate C6, which then activates C7, which then activates C8, which then activates C9. And C9 is going to form a pore. And essentially this pore is going to be inserted onto the membrane of those microorganisms. So imagine you're poking holes on the surface of this microorganism. And of course, water is either going to rush in or rush out, induces a massive osmotic effect, and that microorganism will die. Okay? So C5b, C6, C7, C8, and C9 together form what we call the membrane attack complex, which then pokes holes in the bacteria. So here's your mast cell. So here's C3A, here's C5A binding to the receptors on the mast cells to induce what we call mast cell degranulation. So basically all the granules that contain histamine will be massively released from this mast cell to induce massive vasodilation throughout the body, which essentially means an increase in inflammation that's happening inside the body. Okay? C5A by itself acts as a chemotactic compound, which then attracts the neutrophils, the monocytes slash macrophages to whatever that area of the body is that has that infection. And C5b, C6, C7, C8, and C9 basically forms holes onto this bacteria. So imagine this is an intact bacteria. This is after the membrane attack complex has been placed onto the surface of that bacteria, causing that bacteria to basically break apart. Okay? The overall result of the complex system is what we call oil cans. That's the mnemonic for that. O stands for opsonization. Opsonization refers to the fact that there's a compound called C3b binds to the surface of the bacteria, make the bacteria bigger and more appetizing for the phagocytes, for the neutrophils and the macrophages to come in, bind to it, and destroy it and eat it up. Okay? I stands for inflammation. This is essentially the activation of the mast cell through the binding of C3A and C5A. L stands for lysis. This is formation of the membrane attack complex. That's C5b, C6, C7, C8, and C9 coming together. C stands for chemotaxis. That's C5a by itself, which basically attract the neutrophils and monocytes slash macrophages to the infected site. A stands for agglutination, and agglutination is essentially clumping, or so some of these complement proteins allow for these microorganisms to basically clump together so that when the phagocytes get there, they don't have to really go far to eat up all of those pathogens, right? And then N stands for neutralization, because one of the things that you guys will learn later on in microbiology, if you haven't learned it yet, is that in order for any microorganisms to induce infection of the body, they have to first bind tightly to the surface of your cells and tissue. So therefore, one of the ways to help neutralize that activities of your microorganism is some of these subunits of the complements binding to the surface of your bacteria or your microorganism. If you take away the binding ability of these microorganisms, you essentially have neutralized their infectivity. Does that make sense? Okay. So oil-can, that's the mnemonic for the activities or the overall results of your complement system. Okay. All right. The next thing that the innate immunity also produce is what we call interferons. Interferons are these particular compounds that are made by normal cells of the body and also white blood cells of the body. So there's two different types of interferon. There's type 1 versus type 2. For type 1, you have alpha versus beta interferon. Okay, both of these are secreted by the cells of the body. And the cells of the body will only make and secrete these interferon alpha and beta when they are infected with a virus. Okay, so when you see interferon floating around inside your bloodstream, it means you have some kind of viral infection that's also going on in the body. So imagine if you're a cell, a normal cell in the body, the virus infects you, gets inside the cell, because that's what viruses do, right? Viruses have to get into the cell to basically hijack the cell and turn the cell into a virus-producing factory. That's how viruses induce infection. So therefore, as the virus enters the cell realizes it's infected, it starts to quickly make interferon alpha and beta. The interferon alpha and beta will then be released by that infected cell, which then binds to neighboring healthy cells and stimulate those healthy cells to start producing a protein we call antiviral protein. Okay? So essentially, imagine you're infected, and now you're like, you know what? There's nothing the immune system can do for me. I'm already infected, but I'm a good neighbor, so I'm going to help my neighboring cells and tell them that there's a viral infection that's going on. So I'm going to make these alpha and beta interferons and secrete that out. And my neighbors will pick up the alpha and beta interferons, realizing that, oh, you know what, there's a viral infection going on. I better do something to protect myself now. So these healthy cells starts to make what we call antiviral proteins. Okay? Now, the infected cells eventually gets taken over completely by the virus and starts to produce hundreds of millions of copies of that same virus. The virus eventually breaks out of that cell, killing that infected cell, and starts to infect neighboring cells. There's nothing that these other cells can do to stop the penetration of the virus into these cells. However, these neighboring cells has already been warned ahead of time, right? So they've had a bunch of antiviral proteins just sitting inside the cytoplasm, getting ready. Once the virus gets in and starts to take over, as the virus starts to replicate itself and make its own proteins and stuff like that, the antiviral protein will inhibit those activities. Okay? So if the cells cannot replicate, or if the virus cannot replicate inside the cell, the virus dies. Because viruses cannot survive on their own. They're not cellular structure. Does that make sense? They don't have the necessary components to essentially go through all the protein synthesis and stuff like that our cells do. So viruses needs to get into a eukaryotic cell or a prokaryotic cell, doesn't really matter. They have to get into a living cell so that they can take over all of the internal mechanisms of cellular respiration, making ATP, making proteins, making whatever it is that the virus needs, right? And so therefore, as a virus hijacks a living entity, one of the ways to stop it is to have these interferons stimulate those healthy cells to make what we call antiviral proteins, okay? So that's how interferon works, and it works very, very well, okay? Oftentimes, you're not gonna be sick for most of the time, even if there's like some kind of weird, like flu-like viral infection going on or whatever the case is. Most of us may not get sick unless it's super virulent and it spreads super quickly. But for the most part, if you don't get sick, it's due in part thanks to your interferon. Now for the type 2 interferon, this is called gamma interferon. Anytime you hear the word gamma, think white blood cell. Okay, so the gamma interferon are only secreted out by your white blood cell and by a very specific type of white blood cells, which we'll talk about later on. Here's a picture showing interferon is a cell of the body that is infected with a virus. Here's a virus coming in, right? Penetrating into this particular cell. The cell knows it's going to be infected. There's really not much we can do for this cell. So it quickly makes alpha and beta interferon, secretes it out, binds to neighboring cells that are healthy cells, stimulate these neighboring cells to quickly make what we call antiviral proteins. So therefore, eventually, this infected cells become overwhelmed by that virus. New viral particles exit this cell and infect neighboring cells. Once the new viral particles enter this neighboring cells and tries to do the same thing here, try and make a bunch of like viral proteins or whatever the case is, you already have antiviral proteins ready to inhibit that process. So if you inhibit that process adequately, then the virus cannot make copies of itself. If it can't make copies of itself, it cannot spread the infection to other cells of the body. And because of that, the infection stops. Okay? And that's why you don't get sick, due to the fact that these interferons are working very well. All right, good. Any questions about the innate immune system? Okay. Let's jump into the acquired, specific, or adaptive immune system. Now, think about the term that is utilized to indicate the activities of this immune system. It is a specific immunity, which means it has a very specific target, right? For the innate, it's non-specific, so it doesn't really care what it is. All it cares is that you are not part of me, I'm going to destroy you. That's all it cares about. For the specific immunity, it is also referred to as the acquired or adaptive immunity, which means that once the body is infected, this immune system understand that yes, this virus or this bacteria or whatever this microorganism is the main thing that is causing this infection in the body, then it starts to adapt so that it can recognize this specific microorganism, so that it can focus all of its energy and all of its attack onto one primary target. That's it. All right? It doesn't care if there's like 1,000 different viruses going on. If one is causing the most issue of the body, that's the one it wants to deal with. Okay? And it does it so wonderfully and very, very effectively that oftentimes, once you activate this acquired or specific or adaptive immunity, you can eradicate that microorganism from the body. Okay? For the adaptive immunity, the two types of cells that regulate this will be your T cells and your B cells. Okay? You guys remember these all come from, these are lymphocytes, so therefore they are derived from your lymphoblasts and stuff like that. Now, before the B cells and T cells become fully active, fully mature so that they can roam around in the circulation, or that they can reside in what we call secondary lymphoid organs, which is like your lymph nodes and stuff like that, right? Before that, they have to go through very strict examination. So we call this the immune tolerance exam. For the immune tolerance exam, there's two different stages for this exam. One is called central tolerance, and the other one is called peripheral tolerance. So for central tolerance, imagine if you have a bunch of these immature white blood cells, okay? Immature white blood cells, B cells and T cells, as they develop. Now, both the T cells and B cells are made in the red bone marrow. But as they go through the central tolerance, the immature B cell stays inside the red bone marrow. Whereas the immature T cells will move all the way into what we call the thymus gland. So for the central tolerance exam, the immature B cells are inside the red bone marrow, whereas the immature T cells are inside your thymus gland. Okay? So for this particular central tolerance exam, basically what happens is that as these immature cells are there, a bunch of self-proteins are now being exposed to these particular immature B cells and T cells. So imagine if you guys are all immature B cells and T cells, right? In front of you, that's just a table. and across the table, here comes a bunch of self-protein. Proteins that are made by your body, by all the cells of your body. So at any given time, if any of these immature cells react against these proteins, they are immediately killed off. Make sense? Okay, don't pick up that food, otherwise we're gonna have to kill you. So you better not recognize any of these proteins. Does it make sense, right? This is your immune system. This is a specific division of your immune system. So if you recognize your own proteins, that means you will react against that protein. And if you react against that protein, you will kill your own host, right? So you better not react against that protein. So therefore, during this particular time, the central tolerance, all the immature T cells and B cells are exposed to virtually every single self-protein of the body. If at any time any of these cells react against these, then they die, right? They get destroyed. If they don't react against any of the cell proteins, they will be selected for. Okay? As they're being selected for, they will now be stimulated to become a mature, immunocompetent lymphocyte. Okay? Keep in mind that these mature, immunocompetent lymphocytes basically means that they are now ready to respond against a pathogen that they've never seen before. So therefore, they're also naive in a sense. Does that make sense? Just because you're 18, you leave the house doesn't mean you're experienced. You're gonna get conned into losing money, you're gonna, all this other stuff happens to you. It's called life. So same thing with these particular B cells and T cells, right? Imagine they are now mature because they've passed that central tolerance exam and they become immunocompetent, which means now they have the ability to recognize and react and fight off and kill off pathogens specifically. But they are also naive because they've never seen these pathogens before. So now they will have to react against something that they've never seen before in their life, okay? But there's a way for them to recognize that. All right, so this central tolerance, all of this that I just described, we call that negative selection. Okay, so just kind of add that in if you guys want to. All this activities, all this central tolerance, where the T cells and B cells are exposed to every single self-proteins, and if they don't react against any of those cell proteins, they will be selected for. So essentially, think about this. If the cell positively identify those proteins, they will be killed. If they do not identify or recognize those proteins, that means they have a negative response to those proteins, then they will be selected for. So we call this negative selection. All right. Now, imagine all of these mature immunocompetent lymphocytes, B cells and T cells alike, right? Once they reach this mature immunocompetent stage, the T cells will be released out into the circulation from the thymus gland. The B cell will be released out into the circulation from the red bone marrow. Most of those mature B cells are found within your secondary lymphoid organs, lymph nodes and things like that. Lymph nodes, tonsils, whatever you guys have, right? And then most of the mature T cells are actually found within the circulation itself, okay? However, throughout their lifetime, they are constantly challenged with self-proteins once again. Just because you pass negative selection doesn't mean you are now exam-free for the rest of your life, okay? So constantly, during this mature stage of the B cells and T cells, they're constantly challenged again, over and over again, with self-proteins. So imagine the T cells floating around or rolling around inside the circulation, and it comes across some kind of antigen, and if that antigen happens to be your self-protein, it better not react against that. If it does, for some reason, starts to react against that self-protein, it will also be killed off, okay? B cells, the same thing. If your mature B cells inside your secondary lymphoid organs are exposed to now self-protein once again, if at any time they start to react against those self-proteins, they will also be killed off, okay? And the way we kill off these cells is to stimulate them to go through apoptosis. Another way to really kill these guys is to basically inactivate them permanently. So these cells may become what we call anergic, which means they're like zombie cells, just kind of float around until they reach the end of their life cycle and then they get destroyed by the macrophages. Okay? That type of exam we call the peripheral tolerance. Okay, so central tolerance is essentially a negative selection for immature B cells and T cells. Peripheral tolerance is another negative selection. Well, we don't really call it negative selection anymore. We just want to make sure that these cells do not react to any of the cell proteins. So therefore, peripheral tolerance is for your mature immunocompetent B cells and T cells. Okay? Again, we don't want at any time throughout the life cycle of these particular cells, we don't want any of them to start reacting against your self-protein. Once these cells become activated by a protein, by any protein, hopefully it's a pathogen protein, right, a foreign protein, they will retain the memory of that protein because this is your specific adaptive immune system. So once they basically retain the memory of that protein, they will continue to destroy anything that is attached to that protein. If that protein happens to be a self-protein, a protein made by your cells, then it will destroy any cell that has those proteins. Okay? Imagine if those are your cardiac muscle cells that express those proteins. Well, game over for your heart. Because it will try to destroy every single cardiac tissue that you have. Okay? Imagine if some of these particular proteins are expressed on your connective tissue. Where's our connective tissue located? Everywhere. So therefore, if these particular white blood cells all of a sudden become activated against your own, let's say, proteins on your connective tissue, then all your connective tissue will be destroyed, okay, to a certain extent. Obviously, as these white blood cells destroy them, you try to repair them, but eventually you're going to lose that fight. Because these cells will continue to do their job until that protein has been completely eradicated from the body. That's the intention. Yes?
So how do autoimmune disorders occur? Does that mean that they're passing through central or peripheral tolerance somehow?
Somebody slipped through, right? Or something happened along the way, right? So with, let's say, viral infections, oftentimes when the virus infects your cell, they also take a part of your own protein and they put it on their envelope. So sort of like a masking effect, right? So if they do that, now of course they don't do it like completely because they'll try to take some of these proteins or they'll try to remake some of these proteins to mimic them, but there's some alteration to it. So the vast majority of it still looks like your own protein, but it's actually a viral protein now. So now all of a sudden your white blood cell starts to seize that and say, wait a minute, I know I'm not supposed to recognize this, but there's some differences in the molecular patterns of this thing. But it doesn't matter, the vast majority of it is still the same, but because it's different, your white blood cell responds to it. And therefore, it will look for anything that looks like that protein and destroy it. So if your cells and tissue of the body still makes those proteins, it'll still recognize parts of it and will destroy whatever tissue is associated with it. And that's basically the start of what we call an autoimmune disease. Right? Like lupus is basically a connective tissue disease. Those white blood cells releases antibodies to destroy your connective tissue. Okay, so anyway. That's peripheral tolerance. Now, for the acquired adaptive or specific immunity, there's actually two divisions of it. First one is called the humoral or antibody-mediated immunity. We talked about this already, right? Last week. We talked about how we activate the B cells and stuff like that, right? So we're not going to really spend that much time talking about it. I'm just going to quickly go through it again just to kind of remind you guys of the overall activities of how we activate the B cells. What we're going to talk mainly about today is the cell-mediated immunity, which is a more complex way to activate the T cells. Okay? So here's a table that differentiates B cells versus T cells. You guys can use that table, read up on it. All right, so you guys remember the humoral or antibody mediate, you have a bunch of B cells. They have a bunch of receptors or antibodies placed as receptors on the membrane, right? Keep in mind that these antibodies have antigen binding sites. And because these antibodies expressed by these B cells have never seen this pathogen before, so they basically, each individual B cells will express a type of antibody that may recognize any potential pathogen that may exist in this known world to a certain degree of specificity. Once they recognize it to a high degree of specificity, then they will re-edit some of that particular antigen binding site to now recognize it with a much, much higher, like 100% specificity, okay? But imagine there's some varying degree of specificity for any antigen that you know or that exists, potentially exists in this world, okay? So imagine here's your B cell, and these B cells happen to recognize these antigens to about 70% specificity. Right? Never 100%. I mean, very rare that it's 100% because it's never seen these guys before. So once the antigen binds to the antibodies, this will stimulate this B cell to go through what we call clonal selection. In clonal selection, it's a massive mitotic activities that basically allow for this cell to divide into hundreds of millions of copies of itself. Some of those copies become what we call memory B cells. Some of those copies become activated B cells, which we call the plasma cells, okay? And the plasma cells now will start to make antibodies and secrete those antibodies, and the antigen binding sites on those antibodies will recognize those antigens to 100% specificity, okay? And they release all that out, and they sit there and they just pump out those antibodies like crazy until they die. Now, you guys recall the structure of the antibody. It has what we call heavy chain, so 2 heavy chains and 2 light chains, okay? And in this segment of the heavy chain here, the tip of the heavy chain and the light chain has this portion called the antigen binding sites. So therefore, together, we call that the variable region of the antibodies. The variable region also have another designation called the FAB, which is the antigen binding fragment of the antibodies. Okay. The rest of the antibody structures basically make up what we call the constant region. The constant region is known as the FC, which stands for the constant fragment. Okay. So the variable region is basically going to allow for that antibodies to bind to highly different but highly specific type of antigen, whereas the constant region will differentiate the antibodies into 5 different classes of antibodies. And those 5 different classes of antibodies are listed here. So IgM, IgA, IgD, IgG, and IgE. IgM is a pentamer. It looks like this. It's a pretty large antibody structure. 5 antibodies all bound together, right? Forms a pentamer. So basically it has like 10 antigen binding sites combined, okay? So huge, huge amount of antigen binding sites on this thing. So therefore, it is also the first type of antibody secreted out by your plasma cells during what we call the initial exposure to a pathogen. Okay. Once that happens, you can see that this pentamer, this IgM, is going to effectively pick up or bind to those pathogens and cause them to all clump together. We call that agglutination, right? Cause them to basically stay there, so therefore the phagocytes can come in, the neutrophils and macrophages can come in and destroy whatever is associated with it, okay? Now, the IgM will also induce things like cell lysis as well, okay? IgA is what we call a dimer. It has 2 antibodies bound together and is present in high concentration in bodily secretions, namely mom's milk, right? So the breast milk that is secreted by the new mom to feed, her new child, the newborn and stuff like that is actually very important because not only does it have a lot of nutrients, it has a very high concentration of IgA, which is going to offer temporary protection to that baby for at least the first 6 months or so before the baby's immune system has a chance to basically ramp up and start become activated. IgD is a monomer, it looks like this, and IgD can be secreted to some degree, but for the most part, IgD serve as the surface antibodies on those B cells. IgG is the most common and the most abundant immunoglobulin in the circulation. This is what IgG looks like. It is very good in promoting phagocytosis, This is the only antibody that can actually activate the complement system that we just talked about. Okay. Now, IgG is also the only antibody that can cross that placenta. And oftentimes it will, because that IgG will cross the placenta to protect the fetus while the fetus is still growing inside the mom's body. Okay? But of course, the one IgG that we talked about was the one that basically is sensitized for the D antigen, right? So therefore, you have to match that. You have to type the patient's blood correctly so that you can prevent the mom from making this IgG against the D antigen. Okay? But otherwise, all the other IgG that the mom has is basically going to cross the placenta into the baby and therefore protect that baby from any kind of invading pathogen that may have crossed that placenta as well. All right, and then lastly, IgE is also a monomer, and IgE is basically very, very important to help with the activities to fight off things like parasitic infection and therefore also fight off allergens. So it helps increase the activities of your eosinophils and all the other stuff too. Okay? All right, that's the explanation. Okay, so that takes care of your B cell, antibody mediated or humoral mediated immunity. Okay? The next type of immune activities within the acquired, specific, or adaptive immunity is called cell-mediated immunity. In cell-mediated immunity, this is mediated by the T cells. Now, with the T cells, they don't go out there and pick up antigens by themselves. For the T cell, they have to bind to some kind of protein, surface protein, that is made by your cells, the cells of your body. This is how they respond effectively, right? So therefore, there's different subclasses of T cells. The first one is called helper T cells, and the helper T cells has these specialized surface proteins called CD4, okay? And the CD4 is very unique to the helper T cells, and that's the reason why oftentimes we call the helper T cells the CD4 cells. Now, the function of the helper T cell is that once they become activated for a specific antigen, they will now secrete a huge amount of cytokines, of signals. And some of those signals are vital because it enhances the fighting ability, the eating ability of your innate cells, but also in essence stimulate the B cells to basically release a huge amount of antibodies as well against that specific pathogen. So that's the helper T cells. The second subset of T cells we call the cytotoxic T cells. Cytotoxic T cells contain a surface protein marker called CD8. And it's unique only to the cytotoxic T cells, so oftentimes we will call these cells the CD8 cells as well. Okay? Now the cytotoxic T cells, their function is that they need specific instruction and activation from your helper T cells, mainly. There's different ways to activate the cytotoxic T cells, but one of the main ways, the most effective way, is for the cytotoxic T cells to become activated by your helper T cells. Once the cytotoxic T cells become fully activated, they will now seek out, roam around the circulation, and seek out any cells of the body that is infected by that specific pathogen, bind to it, and eradicate that pathogen. Remember, these guys don't seek out the pathogens. So therefore, the pathogen has to be presented to them in some way, shape, or form. And we'll talk about how that's done in a second. All right, the third type of T cells we call the regulatory T cells, or the Treg. Regulatory T cells were once upon a time helper T cells, but has now retired. So therefore, in order for us not to mix them up, right? Because they both have the CD4 surface proteins. The regulatory T cells now also express CD25. Okay, so both CD4 and CD25 on the surface basically makes this a regulatory T cells. The function of the regulatory T cells is to turn off the activities of the active helper T cells and the active cytotoxic T cells. Right? That's how we regulate these activities all together. All right, now because this is called the cell-mediated immunity, right? These T cells that we just talked about must have some kind of direct interaction they have to be presented this antigen. They're not gonna go out there and seek out the antigens themselves. Make sense? So there has to be something that brings the antigen to them in order for them to become activated, okay? So that thing that basically presents the antigen to these T cells are known as your antigen presenting cells. Okay, so the antigen presenting cells of the body are known as your macrophages and your B cells. Okay? These macrophages and B cells have the ability to take in the antigen or take in the pathogen, break it down, and select out a piece of protein that is unique to that pathogen and present it on its surface. Once it presents it on its surface, it can now bind to the T cells and activate that T cell for that specific type of antigen. Okay? So it's a very complicated and highly regulated pathway because you don't want to inadvertently activate these T cells for anything. Okay? Now, in order for the T cells to recognize these antigens, they have to use their own surface proteins as well. Right? So the surface proteins utilized by the T cells, so in order for them to become active is known as the T cell receptors. Okay? In addition to the T cell receptors, they also utilize the CD4 or CD8 surface marker that's unique to them to also reinforce the binding to your antigen presenting cell. Now, on your antigen presenting cell, there is a surface protein that is utilized by the antigen presenting cell to present the antigen on its surface so that it can fully engage with those T cells to activate the T cells. And the proteins on the antigen presenting cells that does that is known as your major histocompatibility complexes, okay, or your MHCs. For the MHCs, there's two different types. There's type 1 versus type 2 MHC. Type 1 MHC is found on virtually any cell of the body, except for the red blood cell. Okay? Now, for the cell to express MHC1, if it's not infected, if it's a healthy cell, there's no need for that cell to spend the energy to make MHC1 proteins and place it on its surface. When the cell is infected, that's when the cell starts to quickly make the MHC1 and stick that antigen onto MHC1 and place it on its surface to basically raise a flag to the rest of the immune system and says that, hey, I'm infected, come and help me, okay? So therefore, a cell will only produce MHC1 if and only if it's infected with something. Does that make sense? Now, why is it, do you think, that the red blood cells cannot make this MHC1?
They don't have any organelles.
Don't have what?
Organelles.
Which organelle?
They don't have any
Which one's more specific?
nucleus.
They don't have a nucleus. Good. If they don't have nucleus, what happens to the DNA? They don't have any DNA. All their genes are now gone, right? So if their genes are gone, the genes that express the MHC1 is no longer there. So therefore, if a red blood cells become infected, how does it tell the immune system that it is infected? It simply can't. So if your red blood cells is infected, and there's a lot of protozoa, there's a lot of microorganisms out there that love to infect the red blood cell, which is sucks. I mean, it sucks for them because they can't really do anything, right? Once they get into the cell, they're like, well, shit, you don't do anything. You transfer oxygen and CO2. You don't even have any genes. You're not making any protein. What the hell? It doesn't matter. We're already here. We're just gonna replicate by ourselves, right? It's just basically a temporary housing for these protozoa or these microorganisms as they hide inside the red blood cell. Because once they're hiding inside the red blood cells, your immune system won't even see them at all, okay? Which is a problem. But it's not too bad because it is bad because it will eventually destroy your red blood cells, but it doesn't really replicate it massively in that way because the red blood cells don't have the overall, mechanism to make a bunch of proteins and stuff like that. So, pros and cons. Anyway, so keep that in mind, okay? Now, for MHC2, MAC2 is only present on your antigen presenting cells, so therefore only presence on the macrophages and your B cells. Okay. Now here's MAC1 over here on the left. MAC1 looks kind of like this. It has like this 2 chains over here, right? And it has this little bend on the top. We call this the peptide binding cleft or the antigen binding site. So therefore, imagine if there was a piece of antigen. That piece of antigen, oh wait, that piece of antigen will be placed right in that cleft, like that. Okay, this is the antigen. This is the protein, foreign protein structure that is processed by those cells and placed onto that surface. That's the antigen. Over here, this is class 2 MAC. You see now it has 2 separate chains, 2 even chain. And there's also this, basically this area for it to place the antigen here as well. Okay, so that's also the antigen over there. Okay, so keep in mind, MAC2 is only made by your antigen presenting cell. MAC1 is made by virtually any cell of the body except for the red blood cells, and only assuming if that cell is infected. So therefore, do you think the macrophages and the B cell can also make MAC1? Yeah. Any cell of the body, doesn't matter what it is, even white blood cells can also become infected. Make sense? Doctors get sick. Soldiers get killed. I mean, just because you're a soldier doesn't mean you're invincible. Just because you're a doctor doesn't mean you're immune to like all sickness. If anything, you get sick the most. Healthcare professionals, you're at the forefront. You get exposed to every single conceivable pathogens known in this universe, right? So therefore, you're gonna get sick a lot. Just take good care of yourself. All right, so how do these things work? Oh, and another thing I want to mention, go ahead and put this down as well. MAC1 also has an area somewhere around here that allows it to bind to only the CD8 protein. Okay. MAC2 has a binding portion somewhere around here that allows it to only bind to the CD4 protein. Okay. And what do we say about the CD4 versus CD8? The CD4 proteins are only unique to your helper T cells. And of course, regulatory T cells. But now, just leave them out of the discussion for now. So for the helper T cell, it can only recognize MAC2. Okay? And because CD8 is on the cytotoxic T cells, the cytotoxic T cells can only bind to MAC1. Does that make sense? All right. Let's draw the scenario out. So, what happens when a pathogen invades your body? All right, so there's your pathogen. And even if it invades your body and it doesn't do much harm, it's not a big deal, right? Your innate system can take care of it. But let's say if your innate immune system cannot take care of it, and it's now, reproducing massively, overwhelming that innate system, causing a lot of issues like fever and stuff like that in your body, well then you better activate your acquired immunity, okay? So first thing that comes across this pathogen, let's just say you have a macrophage. Okay, this macrophage happens to be your antigen presenting cell. APC. Okay, the antigen presenting cell is going to recognize this pathogen and phagocytose it. How does the macrophage phagocytose this pathogen? Through what kind of recognition? Does it have to bind to the surface of that pathogen? How does it know it's a foreign substance? about this last time. Yeah, because on this pathogen are a bunch of surface proteins, right? And let's say those surface proteins are here, we call them the PAMPs, the pathogen-associated molecular pattern. Now, what is this antigen presenting cell, or this macrophage? What is it used to bind to the PAMP? The toll-like receptor. So imagine this thing is extending upward using the toll-like receptors to bind to the PAMPs. Okay? Once it binds to the PAMP successfully, it will now extend out the pseudopods and engulf that particular pathogen. Okay? Now, once this pathogen is internalized, it will now be packaged inside some kind of vesicle. And eventually it gets broken down, right, through the fusion of the lysosomes and stuff like that. And it gets broken down into these antigens. So it selects out these specific proteins on the surface of this pathogen. And it will present this protein, this antigen, on its surface by placing it on what we call an MHC. So here you have the MHC2 and the antigen complex. Okay, so we'll call this MHC2 antigen complex. Here. So let's color code this a little bit. Now, in addition to placing the antigen onto MHC2, this antigen presenting cell will also place this antigen on MHC1. So we'll call this the MHC1 antigen complex. Any questions? So straightforward, right? Antigen presenting cell, in this case a macrophage, will endocytose or phagocytose that pathogen, break it down, select out the specific antigen that will identify that pathogen specifically, place that antigen on both MHC1 and MHC2 to form these MHC1/antigen complex and MHC2/antigen complex. Now the antigen has been adequately presented on the antigen presenting cell. Make sense? Right, that's why it's called that. After that has happened, this macrophage, let's say if this thing was in your tissue and recognizes pathogen, so now it's gonna leave that tissue, gets back into the circulation, and whilst doing so, it's going to release a type of cytokine. So it'll secrete what we call interleukin 1. Okay? So this antigen presenting cells, once it has phagocytosed and presented the antigens on both MHC1 and MHC2, it will now leave that tissue, moves into the blood, and secrete interleukin 1. This interleukin 1 is going to attract both helper T cells and cytotoxic T cells. Okay, so it's going to attract a huge amount of T cells to this antigen presenting cells. Now keep in mind that every single T cell has something that it can recognize. even if it has never seen that thing before. Okay? So you're gonna have a wide variety of T cells coming in and trying to interact with this antigen presenting cell. But only one will basically become activated. Because you don't want to activate a huge amount of T cells for no apparent reason. Okay? Okay, so this is what happens with the helper T cells once it interacts with this antigen presenting cell. So imagine this MHC2 antigen complex is right here. Interleukin 1 is secreted out, attracts the helper T cells. The helper T cells come and interact with this particular antigen presenting cell. So here's a helper T cell. So helper T cells comes in, and in order for the helper T cells to interact with this complex, it has its own receptors as well. And the receptors that allows it to interact with MHC is called the toll-like. Oh, sorry, the T cell receptor. Okay, not the toll-like receptor. All right. Now imagine if these T cell receptors, these TCRs, on these particular T cells, if this particular helper T cell was basically produced and it has this portion right here that will recognize the antigen. But if this portion right here cannot recognize this antigen to a high degree of specification, then this helper T cell will disengage and leave. Okay? Because it doesn't have the correct antigen specific, like the specific antigen recognition site. Okay? So multiple helper T cells will come in and do that until you get a helper T cell that has this pattern. Okay? So this pattern will recognize that antigen to at least 70% specificity. Okay? So some higher degree of specificity and stuff like that. Now, once it has achieved the recognition of that antigen, well, first of all, the TCR has to recognize the MHC2, and then this portion on the TCR must recognize that antigen. To reinforce this interaction, on the helper T cell, you also have that surface protein called CD4. So CD4 will also bind to MHC2. Okay, because remember, on the MHC2, there is a specific binding site for CD4. Okay, now once this occurs, once the CD4 binds strongly to the MHC2, this cell will become stimulated internally. As it becomes stimulated, it will now start to make a new surface marker, and this surface marker is called, we'll spell it out, interleukin-2 receptor. Okay? So the interleukin-2 receptors are now expressed on the surface of this helper T cell. This helper T cell will also secrete a bunch of interleukin-2. So interleukin-2 looks like this, or just make it look like that. Who knows what it looks like? Who cares? Right. So in addition to expressing interleukin-2 receptors, it also secretes interleukin-2. Make sense? Now guess what the interleukin-2 does with the interleukin-2 receptors? So interleukin-2 binds to interleukin-2 receptors. Okay. Once interleukin-2 binds to the interleukin-2 receptor, this is now an autocrine process, so like a self-stimulation process. That this cell, this helper T cell, will now go through massive activation. And that massive activation is referred to as clonal selection, once again. Okay. So it's a complicated process of multiple regulatory pathways to ensure that if you want to really activate this helper T cells, it needs to go through all of these steps so that it can be activated. Otherwise, it will not be activated. If this helper T cell for some reason was activated inadvertently, then it will not do some of this stuff and it will become anergic. It will become a zombie cell for the rest of its life. Because if that's the case, right, let's say if this antigen wasn't a pathogen, but it was like part of your self-protein, and let's say if this helper T cells all of a sudden start to react against that self-protein, it will become anergic. That's what we call peripheral tolerance. Okay? But in this particular scenario, that pathogen is a true pathogen. So therefore, this helper T cells will bind to recognize that antigen. CD4 reinforces that binding, stimulates it to release interleukin-2, express interleukin-2 receptors, and essentially now it's an autocrine process. So it's an autoregulatory process as interleukin-2 binds to the interleukin-2 receptor to stimulate clonal selection of this individual helper T cell. So only one helper T cell gets activated. Okay? We're not going to activate all gazillion helper T cells that you have in the body. You only want to activate one helper T cells because this helper T cells recognizes this antigen specifically. Okay? Now, in this clonal selection, what happens to this helper T cell is that some of them become memory helper T cells. Okay. The other become effector or activated helper T cells. Okay. So the memory helper T cells retain the memory for that antigen. The effector or active helper T cells are now active. Okay. So it's going to do something. So effector also referred to as the active helper T cells. Now remember, I told you guys, the helper T cells, their job is to secrete cytokines, secrete some kind of signals to massively activate your immune system. So one of the things that it does secrete is this substance. So here, it secretes multiple. So it'll secrete what we call tumor necrosis factor alpha. That's just a short list, right? There's a bigger list than that, so we'll just write this out. Which is known as TNF alpha. Okay? And tumor necrosis factor alpha is going to activate, enhance the activities of your neutrophils and your macrophages mainly. Okay? So TNF alpha increases activities of, we'll just call it innate cells. So any innate cells that you can think of. Neutrophils mainly, macrophages, maybe eosinophils, depends on what kind of pathogen you have. Let's say you have a parasitic infection, right? Then you want eosinophils to have an increase in activity so it can release more enzymes, break down the worms, kill the worm better, okay? If you have some kind of allergic reaction, it's gonna quickly neutralize that allergens and therefore prevent you to have massive particular, reaction from that, okay? So, increased activities of your innate cells. That's the job of TNF alpha. Okay. The effector helper T cells also secrete what we call interferon gamma. Interferon gamma is going to increase the activities of your B cells so that the B cells can now secrete huge amount of antibodies. Okay. So increased activities of antibodies will add to of B cells to increase antibody secretion. Yes? Okay. All right. A third substance that is secreted by these effector helper T cells is the substance we call granulocyte monocytes. Stimulating factors, which stands for, or abbreviated as GM-CSF. Okay. GM-CSF will stimulate the red bone marrow to increase the production of what? Of all of your granular leukocytes. Neutrophils, eosinophils, basophils, right? And monocytes. Which then, you guys understand, becomes macrophages. Good. So, right? So, this effector helper T cells, in addition to secreting the the tumor necrosis factor alpha to increase the activities of these innate cells, why not stimulate the red bone marrow to make more of these innate cells? So now you have a much bigger amount, larger concentration of these innate cells that has a higher activities overall. So therefore, much more effective response, like phagocytosis or whatever response that you need, right? In addition to that, you also have interferon gamma, which will stimulate your B cells to secrete all forms of antibodies. Depends on which type of pathogens that's infecting your body. Okay? So this is what happens to your helper T cells. This is how you activate the helper T cells. Okay? This is called cell-mediated response. Does that make sense? Because this cell is mediating the response of this cell. This cell, the helper T cells, does not look for the antigen by itself. It's not like the B cells that can bind to the antigen specifically or the pathogen specifically by their antibodies. It's not like it's the macrophage of the neutrophils that recognizes the PAMPs through the toll-like receptors. Okay. So these cells do not go look for those things. They have to be wine and dine a little bit. They have to basically be presented. So think about these helper T cells as the generals, the commanders. You guys ever served in the militaries? Most of those generals and commanders sits in the AC room, air conditioned room. Basically just sit around waiting for somebody to bring them a message. Oh, we're being attacked? Okay, let's just, here you go. Send out the signal, activate the whole cavalry. Okay. Now, if you want to really fight off a specific pathogen, you want to send out your highly trained soldiers. And those highly trained soldiers are your cytotoxic T cells. So how do we activate the cytotoxic T cells? This is just a picture showing the same thing that I just drew out for you guys. So this is how we activate the cytotoxic T cells. So imagine when the helper T cells were being activated. This antigen presenting cell also presented this antigen on MAC1, correct? So therefore, there's two of these particular presentation going on here. Now, of course, the interleukin-1 secreted by this antigen-presenting cell has attracted a huge amount of T cells, both helper and cytotoxic T cells. The cytotoxic T cells will again come down here and interact with this cell, with this particular presentation. Okay, so this is the cytotoxic T cells. All right. Now, these guys will also have T cell receptors that will interact with the MAC1 structure. And it will also have a portion right here that will recognize the antigen. So again, it's going to be a series of these cytotoxic T cells that basically may have a different type of recognition pattern here. Make sense? So let's say if you have this cytotoxic T cells coming in, trying to bind to this antigen, doesn't recognize it, therefore disengages and move on. It's not going to be activated for that. Eventually, you're going to have a cytotoxic T cell that has that pattern. So therefore, on the TCR, recognizes that antigen to a higher degree of specification. Okay, so let's say 70% or something like that. Now, to further strengthen this interaction, to to make sure that this interaction stays locked, on the surface of your cytotoxic T cells, you have the CD8 protein. So the CD8 also bind to the MAC1. Just remember I told you guys on MAC1, there is a specific binding site just for CD8. Okay, so this is CD8. Okay. CD8 is unique to cytotoxic T cells. CD4 is unique to the helper T cells. I'll put an S here just to make sure you guys understand multiple of them comes to interact. Okay, so once the CD8 binds to this MAC1 complex and the T cell has recognized that antigen, this cell will become activated. Okay. Once it becomes activated, this cytotoxic T cell will express interleukin-2 receptor. Spell it out. So it makes a bunch of interleukin-2 receptors, place it on the surface. Okay. Now to ensure that this cytotoxic T cell, basically because this guy is being activated at the same time as the helper T cell, okay? To ensure that this is the cytotoxic T cells that you want to stimulate, in this process, in this pathway, the cytotoxic T cell will only express interleukin-2 receptors. It will not secrete interleukin-2, okay? But it has the interleukin-2 receptors, which means while this is going on, the helper T cells has already secreted a huge amount of interleukin-2. So the interleukin-2 that is coming from your helper T cell will bind to your cytotoxic T cells. Okay. Keep in mind, interleukin-2 not being secreted by those cytotoxic T cells, but these interleukin-2 are secreted by this helper T cell that is being activated at about the same time as this cytotoxic T cell. This ensures that this activation is coordinated. It is activated for the same antigen, not activated separately, because there's oftentimes some process that you can activate this cytotoxic T cell by itself as well. But this is just to ensure that this cytotoxic T cell will be activated for that specific antigen. Okay. All right. Once interleukin-2 from the helper T cell binds to the interleukin-2 receptors on the surface of your cytotoxic T cells, this cell also becomes activated. And as it becomes activated, it will also go through clonal selection. As it goes through clonal selection, it will become memory, cytotoxic T cell, and it will also become effector or active cytotoxic T cells. Okay. Circle these for you guys, so you may want to know these. Any questions? Take a picture of her, Lisa.
So after both helper T cells and the cytotoxic T cells go through the process of the clonal selection, now it will have the 100% specificity.
Yeah. The next time they come across the same pathogen, this process will be much faster. Well, the next time you already have like a bunch of memory B cells that will secrete huge amount of antibodies for you, right? Okay. So therefore, you already have that immediate protection.
Pretty much just like feed it.
Yeah, let's say if that pathogen is so strong that it kind of overwhelms all of those antibodies, then these guys are already ready to just go through clone selection.
Okay.
So the next time this happens again, this process is a lot faster. We're talking about like 10 times faster. Okay. All right, good. Any other questions about this?
And then the effector cytotoxic T cells, they don't secrete anything?
I'll show you what they do. That's why I have to erase, because I need the board. Good? Okay, so keep in mind, helper T cells secrete these things to activate overall immune activities. So toxic T cells, remember, these are your highly trained, let's say, soldiers, right? This is your general, so he's just sending out orders. It's like, oh, you know, National Guard, just get activated to fight off this foreign pathogen, right? Telling these B cells, his whatever, his lieutenants, go ahead and secrete a bunch of antibodies and stuff like that, so then we can protect the body. And it tells the cytotoxic T cells, hey, I'm gonna activate you now. You go out there, secret, go out there, look for all of these particular infections, deal with that. Okay? So, this is how it works. So hopefully in your studies, you should be able to reproduce this. Right? If you can reproduce this, you now have effectively told the same stories that I have. If you can tell the story with all the specific players and characters and their functions and things like that, you now will know the cell-mediated immunity, because that is a huge portion of your lab exam. Well, I wouldn't say huge. All right, so this is what happens to your effector cytotoxic T cell. So imagine you have three scenarios, okay? You have these cells right here. These are the cells of your body. Okay, so here's your cytotoxic T cells. So, effector, cytotoxic, T cells roaming around, they're angry, they're active, they're now looking for that specific target so that they can deal with that target. They come across this first cell. On the surface of this first cell, there is no MAC1. Because remember, cytotoxic T cells can only recognize MAC1, okay? So if this cell does not have any MAC1 antigen complex on its surface, what can you say about this cell? Is it infected or not?
No.
No, okay. So it leaves the cell alone, and it moves on. Comes across this next cell. So this cell has an MAC1 on the surface, and it also has an antigen placed onto that MAC1 as well. Okay. This cytotoxic T cell comes across this guy, recognizes the MAC1 because that's what the TCR on these cytotoxic T cells will recognize. But does not recognize that antigen. So what does that mean? It means that the body is infected with two things. So therefore, this cytotoxic T cell said, hey, you know what, you're infected, I understand. I can't help you, it's not my job, okay? This is not the target I'm looking for. Some other one will come along and help you. Just hang in there, okay? So disengages, moves on. Leaves the cell alone, hopefully the cell gets, resolved in time. Comes across a third cell, or a third set of cell, let's just say. So this cell has the MAC1 on its surface, and it also expresses our favorite antigen, the one that we know. So cytotoxic T cells will come across this cell. Recognize the MAC1 using the TCR. Recognize the antigen because that's what it's activated for, okay? Once it recognizes all that, it further strengthens this with the CD8 protein. Make sense? All right, now, once that happens, this cell becomes responsive and it will secrete what we call perforins. These perforins are substances that will eventually place little channels onto the surface of this infected cell. Okay? And it also secretes another substance, and we call these the granzymes. And the granzyme enter this cell through these channels, and it will help this cell resolve its infection. And the way that this particular cytotoxic T cell helped this cell resolve this infection is to kill it. You're infected? Let me help you. Go kill yourself. And the cell's like, thank you very much for coming to help me. I will do just that. Pop, 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 apoptosis. And it dies. If this cell dies, whatever pathogen is inside the cell also dies. Okay? Because whatever pathogen that needs to get into the cell to replicate needs the cell in order for it to propagate its infection. So if you kill off infected cells of the body, you actually stop the spread of infection throughout the whole body. It's sort of like you have a forest fire and you just burn that rim of fire, basically prevent the spread of that fire, right? So that the fire doesn't spread. Unless the fire has like 100 miles of wind behind it to help it move along. You can't really burn enough, which is what happened down in LA several months ago. Okay, good. Any questions about the activities of your cytotoxic T cells? This is what you train these guys for, right? This is why you activate these cytotoxic T cells so that they can go out and do this job. And they will do it very effectively. They will seek out all of these infected cells and destroy every single cell that is infected with that specific pathogen. And when this is happening, this is when you actually exhibit symptoms of that inflammation that's going on, of that reaction that's going on, that's carried out by your white blood cells. Okay? When the virus or bacteria or the microorganism infect you, you show virtually zero symptoms, unless you have massive cellular destruction, right? From, let's say, a highly virulent type of pathogen. If a pathogen is smart, it does not kill its host quickly. Does that make sense? If it kills the host too quickly, you can't spread the infection. If it wants to live, it needs to basically slowly infect its host so that its host can go out there to the community and spread the infection to everybody else. That's why the flu is very, very highly virulent in that way. Okay, the flu doesn't like kill you immediately, but it kind of, makes you kind of, you weren't really that sick to begin with, or you don't even feel it. You may wake up one day and you're like, I feel like shit. So what? We all feel like shit all the time, right? So you still go to school, you still go to work, and sneeze all over the place, touch everything. Co-workers come by, start touching it, you're like looking over, oh my God, that's a pretty good potato salad. Oh, sorry, like spread the germs onto their food. And now they're consuming it. Like pat them on the back, shake the hand, give them a hug. That's it, that's how you spread the infection. And that's why the hospital is the petri dish for every single microorganisms that you can think of. Right? This is where you also get multi-drug resistance type of microorganisms. That's what, okay, in the hospital. Anyhow, so if think about, put yourself in that pathogen's shoe or membrane or whatever, right? Think about how you want to spread your infection to all the hosts. If you kill the host too quickly, you leave little chance for that host to pass the infection to other hosts, right? Like the Ebola virus once upon a time, right? When the world was much larger and we have isolations throughout the whole world. That's why, let's say the Ebola virus, as it hits these particular tribes in somewhere in Central Africa or whatever the case is, those tribes are isolated from the rest of the population of whatever that country is. So therefore, within several weeks, all of those people died. If they die, the virus has nowhere else to go. So basically, they quarantine themselves. Now the world is much smaller because of international travel, right? So therefore, before we know it, we're gonna have a lot of these particular international infection showing up in our backyard, right? There are what we call multi-drug resistant strains of tuberculism that will actually resist to the mainstream medication that we give for TB. Right? And as healthcare professionals, you are all potentially exposed to some of that stuff as well. Okay? There's something we call, like back in the days, right? We have sexually transmitted infections like chlamydia and gonorrhea, usually easily treated with 1 or 2 pills. Now we have super gonorrhea and super chlamydia, right? Those things are very hard to treat. So once they become multi-drug resistance, they're smart. These particular pathogens, they mutate rapidly. And therefore, they get resistance to a lot of these particular medications all the time. Anyhow, so that's that. Any questions about that? Okay. All right, let's talk about the regulatory T cells and their role in all of this stuff. Just keep in mind the microorganisms will win one day. Just accept it. Right, just enjoy it while it lasts. We're only here for like a short 80 years max. You live longer than that, it's just a waste of space. Just kidding. I'm not saying that out loud. As long as you have quality of life, that's the important thing. All right, anyhow, let's talk about these cells. So the regulatory T cells. So now imagine these cells here, right? The regulatory T cells. Okay. What they do is that they will secrete cytokines or some kind of signals to reduce the activities of your effector T cells. Like if you have an active helper T cells or active cytotoxic T cells, they will do their job to basically bring everything back into order. Okay, so imagine you have an active cytotoxic T cell. The regulatory T cells will secrete a type of interleukin called interleukin 10. Of course, on this particular surface, there's a receptor for that, so interleukin 10 will bind to this active cytotoxic T cell and stimulate the cytotoxic T cell to express a receptor we call a FAS receptor. So expresses a bunch of FAS receptors. Okay. This active cytotoxic T cell will also secrete a bunch of ligands we call the FAS ligands. Ligands are just anything that is secretory from the cell. So the FAS ligands. Okay. The FAS ligand is going to bind to the FAS receptors. And this is how the immune system rewards a job well done. Once the FAS ligand binds to the FAS receptor, it will stimulate this cell to go through apoptosis. Good job on killing or destroying all the virally infected cells or all these infected cells. You resolve the situation, we're now healthy again, go kill yourself. And these guys are like, oh, thank you very much. Popping like flies, right? It's okay, we have the memory of that particular pathogen. Okay? Sometimes it looks for a more effective way to induce apoptosis in these cells. So imagine you have 2 of these cytotoxic T cells. They come in close proximity to each other. Interleukin 10 stimulates both of them. one of these guys will now express fast receptor, the other one will also express fast receptor. And instead of releasing out the fast ligand, these cytotoxic T cells will also express the fast ligand on their receptors. So on this cell, they have the fast ligand. Instead of secreting it out, they can now just place it onto the surface. So therefore, when this cell has their fast ligand binding to the other cell, this caused the other cell to go through apoptosis. And when the fast ligand from this cell binds to the fast receptor on this first cell, it also stimulates this cell to go through apoptosis. It's like dual cell suicide, basically. Just a more effective way to kill off these active cytotoxic T cells. Okay. All right. Any questions?
So regulatory T cells just keep secreting interleukin 10 always throughout the body. Okay.
So as soon as some of these cells go through that clonal selection, well, all depends, right? So as soon as they go through clonal selection, depends on the severity of the infection. The regulatory T cells may wait, or they can start secreting IL-10 right away, or interleukin 10 right away, to start to decrease that number, because those helper T cells may still continue to go through clonal selection and stuff like that too. Depends on the severity of that. So there's always an interplay between how many of these active cells you have and how much you can bring it back down again. Okay. Any questions about that? Okay, good. All right, so let's talk about what happens after all of this is said and done, after the infection has been resolved, or after some kind of protection has been implemented by the body. Now you gain what we call immunity to that pathogen. You are now immune to that pathogen, which means you are now protected from that pathogen ever causing any kind of infection in your body again. Okay? Now, to produce immunity, you either will produce the immunity in what we call an active immunity or a passive immunity. Active immunity refers to the activities or the production of memory cells. So let's say if you've been exposed to a pathogen in some way, shape, or form, and your immune system has now gone through the process of recognizing it, becoming activated, and now has formed memory cells, both memory B cells and memory T cells. Okay? You have now achieved what we call active immunity. If in this protection is a temporary protection where you are given a bunch of antibodies from something, right? Then those antibodies will just bind to the pathogens and destroy the pathogens without allowing your own immune system from going through all of those processes of becoming activated. And therefore, at the end of that, you will not have produced any memory cells. So if you are protected without the production of memory cells, we call that passive immunity. If you are protected with the production of memory cells, then this is called active immunity. For active and passive immunity, there's a natural way to achieve that or to receive that, and there's an artificial way to also receive that. So that's why we're going to break it down into 4 immune statuses of the body. The first one is called natural active immunity. Natural active immunity, again, after this process, you have now achieved the production of memory cell. But you've achieved this by being infected with that pathogen, going through all those stages of infection, getting sick, high fever, sweating, taking days off work, and whatever the case is, or being hospitalized because of the severity of this infection, and now has recovered from that infection. Okay, once you recover from that infection, you have now basically produced adequate amount of memory cells. So how many of you guys have had the chicken pox, the actual chicken pox? Wow, that's more than I thought. Okay, usually it's like 4 out of, whatever. Good. That means that you've gone through, you got infected with that virus, you've gone through the series of 7 to 10 days of, you know, getting super sick, high fever and all that stuff, and then after the fever subsided, you have a bunch of lesions all over your face and body, and you have to wear mittens because it becomes super itchy and you can't scratch yourself, otherwise you're gonna leave massive amount of scar tissue and stuff like that in your body, right? Sometimes you have to bathe yourself in like calamine lotion, this weird pink chalky type of lotion and stuff like that. It's kind of soothing, but it makes you look like a, you know, I don't know, weird little marshmallow and stuff like that, right? But anyhow, at least you recovered, and now you have the memory cells against that particular virus. So the next time you walk into a room and one of your classmates shows signs of the chickenpox, for some reason they didn't stay home, I don't know, maybe they thought it was acne, whatever the case is, or your patients came into your clinic with the chickenpox, you're like, it's okay, give me a hug. You're protected. It's all right. Okay? Because as soon as you come into the same space with that person, it is highly contagious. Well, don't hug your patient if they do it after
They have
chickenpox.
like pus like on
I
their
know.
thing.
It's okay. Little kid crying or whatever. Okay, so that is called natural active immunity. Now, the other type of natural way to become immune to something is what we call natural passive immunity. Now, this is called passive immunity because you don't have the production of memory cells. So for whatever process that's occurring, you are receiving antibodies from some other source outside of your body. But you're receiving this the natural way, okay? Which basically means one of the primary example of that would be receiving or consuming breast milk as a baby. As we're breastfeeding, we're consuming that milk high in calories, high in nutrients, and very high in immunoglobulin class A. So very high amount of antibodies in that milk. The antibodies produced and is in that particular breast milk is now going to protect your body against pathogen. So just in case a pathogen happens to enter that baby's system, the baby's immune system is so weak it will not respond to it. But the IgA that is in the breast milk will bind to and neutralize those pathogens, destroy them, before those pathogens have a chance to induce infection in that child. Make sense? So it is an immunity, a protection. But this protection through the naturally receiving these antibodies is going to just protect your body and prevent massive infection in the body. However, your immune system did not have a chance to react against that pathogen. Because your immune system did not get a chance to react against that pathogen, you will now not produce any memory cells. Okay? So that is called natural passive immunity. Now, the other type of active immunity is what we call artificial active immunity. In artificial active immunity, this is basically akin to getting a vaccination. So it's a prepared, let's say, purified protein or some degree of active, like viruses and stuff like that, that you have what we call attenuated their virulence. So you basically turn off the overall activities of these pathogens, but you still keep them whole. You don't break them apart, and you just kind of place it into a solution and inject that into a patient. Okay. So these are all vaccines, and there's different types of vaccines that is prepared multiple ways. Nowadays, vaccines are a lot more synthesized because they have multiple protein structures that is collected together and injected all together into your body. So therefore, now it gives your immune system a chance to recognize these pathogens without you being sick. So you actually don't have to go through the stages of disease because you're actually not being infected by these pathogens. You're just being introduced to pathogens in their whole form, so your immune system can react against these pathogens. And when they do have a chance to react against these pathogens, they will now produce antibodies against those pathogens. Okay? So if you ever got a vaccination, that's precisely why. Okay? Have you guys ever had the hepatitis B vaccine? Pretty sure, because all you guys will have to have one in order to be healthcare professionals. If you don't get a Hep B vaccine, you may not be able to get a job in the clinic. So let's say you go to get your Hep B vaccine. You guys remember how many shots it takes? 3 shots. Okay. You go to the clinic, they give you a shot that day. You come back in about a month, they give you a second shot. You come back in about 6, 9, or 12 months, depends on whatever it is. So maybe a whole year, you get the third shot. After the third shot, they bring you back again and check your blood. What are they checking for? Antibodies against the hepatitis B. Okay, and it's supposed to be a virus. So now, what if you haven't made any antibodies? What do they do then? Has everybody run into that scenario? Would you guys all got Hep B vaccines and everybody made antibodies, like, you know, up the wazoo? What if a scenario is that you don't make any antibodies? So they give you that series again. They give you one shot that day, they bring you back in 1 month, give you a second shot, bring you back in about 6 to 9 months or whatever, give you the third shot. Okay? After that, they bring you back again and check your blood. What if you still haven't made any antibodies? That's it. They don't give you any more. Okay? They say if the hospital do hire you or you work in a healthcare clinic or whatever the case is, just protect yourself. Because hepatitis B is the most common blood-borne pathogen. If you get a needle stick, 30% chance you're gonna get hepatitis B, right? And 0.3% chance you may get HIV. So therefore, think about the percentage of that. Hepatitis B is one of the most common blood-borne pathogen. Okay, that's what we look for all the time. Anyhow, so that's that. That's artificial active immunity. Okay. Now, artificial passive immunity is basically whatever it is that's happening to you. You're given an injection, a prepared solution that contains a huge amount of antibodies against that specific pathogen. So therefore, let's say you get bitten by a rabid dog, right? And because the virus that is carried in this particular rabid dog, called the rabies virus, basically can be highly virulent. And once it gets into the neurons, that's it. Game over, basically. So therefore, you need to neutralize them right away before they basically move any further and deeper and hide away in your away from your immune system. So therefore, immediately you're given an injection, and then that injection contains a high concentration of antibodies against that pathogen. That is just an immediate protection because your body may not have adequate time to produce antibodies or produce memory, because you may die before the memory is actually produced, right? So therefore, it is something that you need to do rapidly. Like if you get bitten by a snake, okay, you get antivenom, and that antivenom contains a huge amount of antibodies, because oftentimes your body will not have adequate amount of time to produce memory cells against that antibody. Remember, think about this. The first time you get exposed to that venom, it may take you up to a whole month to make antibodies. If that venom is highly dangerous and depends on the type of snake that bites you and the amount of venom that is injected into your body and where they bite you, you may be dead in hours. Okay, so there's absolutely no time to wait for your body to make it, memory from that. So immediately you need that protection. They give you that antibody shot or that PrEP shot, and basically that contains a huge amount of antibodies to protect that pathogen from destroying your cells. Okay? But it doesn't give your immune system a chance to make memory cells. So therefore, you still don't have the memory against whatever that is. That's why snake trainers, they receive a small amount of venom every so often so that they can basically induce that memory cell production in their immune system. When you're bitten by a snake, naturally the snake's not gonna be like, I'll give you a chance to make memory cells. Then come back and fight me again. The snake's like, screw you. You scared me, I'm gonna kill you. All right, so here's a chart showing primary versus secondary responses, or primary or initial versus other exposure. So remember, during the primary response, once your infection or once those pathogen enters your body, it takes up to about 4 weeks in order for you to make adequate amount of antibodies. Okay, once you produce that memory cell, if you're exposed to the same pathogen again, it takes about 2 hours for your memory cells to produce massive amount of antibodies. Okay, what antibodies are produced during the primary response? What class of antibodies would that be? Or IgM, very good. What class of antibodies are produced during the secondary response? IgG. All right. Alright, the rest you guys can read. We're not gonna talk about allergies and sensitivity, and we're not gonna talk about autoimmune diseases. Okay. Any questions? Take a break. We're gonna jump into the next chapter. We have to because we lose Monday.